about some meiosis. So we did mitosis on last week, which mitosis, I told you, and we wrote down some characteristics. Let's just review them briefly, please and thank you. We talked about that mitosis occurs in what type of cells? Somatic cells. Okay, I thought you said genetic. Yes, somatic cells. Prokaryotic or eukaryotic? Eukaryotic. Sidestep, what do prokaryotic cells undergo? Binary fission. Okay? Clear distinction. If I'm replicating somatic cells, let's give some examples of somatic cells really quickly. What are some somatic cells? Skin. Liver, heart, kidney, bone, all of that stuff. Any organs that you have, okay, those are all going through mitosis. Muscle, mitosis. Brain, mitosis. All right? When we talk about mitosis, we want those cells to look the same or different when they're done. The same. In fact, we describe mitosis, the product being two genetically identical daughter cells. Like that's the technical wording for it. Two genetically identical daughter cells. Okay, we want them to look the same, exactly the same. There are 4 stages, 4/5 stages of mitosis. That would be prophase, metaphase, anaphase, telophase. If there's 5, it's prometaphase, right? And I had discussed that. Interphase is the other part of the cell cycle, but it is not part of mitosis. When I was grading your quizzes and it asked for the 4 stages of mitosis, a lot of you put interphase first. It's not right. The cell cycle has interphase, but mitosis does not have interphase. So the interphase is G1, S, G2. Those are the 3 parts of interphase, and many of you got that one right. Okay? So G1 was gap 1, when it's just growing. S was synthesis. It's replicating the DNA. And G2 is preparing for cell division. And we had covered that all last week. So that was kind of a recap of mitosis. Now, last question. Mitosis is regulated by what? What regulates it? What was it called? The physical molecule that regulates it. CDK. CDKs, cyclin-dependent kinases. And I didn't help, but I discussed the fact that it says kinases, so it ends in ASE. It's an enzyme, and all enzymes are proteins, right? So these CDKs regulate that cell cycle and make sure that it goes the way that it's supposed to, and that cell looks the way that it's supposed to look when it gets to various stages, so that there's no mutation. And if there's an issue, it tries to fix it before it lets it go on. Now, when that goes well, all your cells are happy, everything looks good. What happens when those CDKs are inhibited or not working? Cancer, right. And cancer, by definition, is uncontrolled mitosis. And it has no checkpoint. It has nobody regulating, saying, hey, you're not ready. You're not ready for this yet. And the cell continues to go through it, and because it can't grow out like normal, it grows up and it forms tumors. And we discussed that as well when we covered mitosis. Okay? So that was a quick recap of mitosis. The reason why I wanted to make sure that I did that is because today people get this process mixed up with mitosis, because there are a lot of similarities, but big picture, if it says meiosis, I'm dealing with what type of cells? Sperm and egg. So I'm going to write this down. So meiosis. And you are exactly correct. It is sperm and egg, and only sperm and egg. But because we're in science, calling them sperm and egg is too simple. And it's too straightforward. Like anybody can say that. So in science, we refer to sperm and egg as gametes. Sperm and egg are referred to as gametes. And this is across all species. So even plants have gametes. Okay? when we say gametes, that's referencing the reproductive cells of that organism. So for you and I, sperm and egg is spot on. Now, while I have this here, gametes here, I want to put in humans, where are gametes produced? Gametes are produced where do we make sperm and eggs? Where are eggs made? In the ovary. Eggs are made in the ovary, or ovaries, because you have two. Where are sperm made? In the testicles, yes, or the testes. And again, this is too simplified, too simple for what we say in science. So instead of saying ovaries and testes, we say gonads. And I'm saying that because you need to be familiar with these two words. Gametes are produced in the gonads. Gametes are produced in the gonads. Okay, so some takeaways, and of course this is on the intro slide, but it is super important. The end product for meiosis. The end product for meiosis is 4 genetically different daughter cells. 4 genetically different daughter cells. And they only have half as much genetic material as the parent. 4 genetically different daughter cells that contain half the genetic material of the parent. 4 genetically different daughter cells that contain half the genetic material of the parent. And key point right here. Females make all of their eggs before they are even born. All their eggs are stored in their ovaries and they hang out there until she hits puberty. And then once she hits puberty, she ovulates once a month. And the ovaries take turns. Right ovary 1 month, left ovary the next month. And that's how it is in a textbook situation. Of course, there are irregularities in overovulation or no ovulation. For a male, he does not begin to produce sperm until he hits puberty, and he won't stop until he dies. Females, as soon as they are born, they no longer make any eggs. They're stuck with the number of eggs that they have. And they'll ovulate those eggs until they run out, and then they'll hit menopause, and that's it. So menopause is when she ran out of eggs. So females create all of their gametes before birth, and they'll run out during life. Where males begin making their gametes during life, and they don't stop making gametes until they die. Okay? So it's timing as far as successful reproduction. But that's taking us into an anatomy lecture. We use gametes, so sperm or egg, especially when we're referencing humans, for sexual reproduction. And here you see the term gamete used. You need to be familiar with the term gamete. Meiosis produces gametes with half the number of chromosomes. And remember, chromosomes are DNA, and DNA is genetic material. So you're going to hear those words used interchangeably. Chromosomes are DNA, and DNA is genetic material. So don't get yourself confused. Meiosis produces gametes with half the number of chromosomes. That way, and we discussed this last week as well, it has half the number of chromosomes so that when sperm and egg meet, you have a full set, a full number. So for humans, let's use us because it's easiest to learn on yourself. All of our cells have 46 chromosomes, 46, unless It's a gamete, and your gametes then just have 23. So an egg would have 23, and a sperm would have 23. And the science behind that is that if that egg and that sperm fuse to form a baby, which is what we would call fertilization, we now have 46 chromosomes, which is exactly the exact number of chromosomes necessary to create a baby. If you had 46 in a sperm and 46 in an egg, when they fused, you would have 92, and that baby would not live. Humans require 46 chromosomes. You could have 45 and survive, but you would have complications in life. You could have 47 and survive, but you would have complications in life. If you had 49, you would miscarry in utero. You would not make it. You would be selected against. And if you had 41, you would be selected against. Okay? That's natural selection taking place. The term zygote is what we call a baby as soon as fertilization occurs. When sperm and egg meet, you're a zygote. You are one cell. And you stay one cell for about 1 to 3 hours. And then you'll start growing. It is during this time that it determines what eye color, what hair color, how height, your height, not how height. Your height, your weight, your basal metabolic, all of that is determined here. What genes are going to be turned on and off occur right here before your parents even knew that they were pregnant with you. But we have a lecture on that later this week as well. The paternal chromosomes come from the dad. Where do the maternal chromosomes come from? The mom. Okay? The dad determines the sex of the baby every single time. The dad makes male and female sperm. The mom, she only makes female eggs because she only has female chromosomes. So the sex of every child is determined by the father. Okay? It discusses here homologues or homologous chromosomes and alleles, and we're going to get into those genetic terms this week as well. Okay? But homologues or homologous chromosomes, so you have a cell, and you'll get chromosome 1 from mom and chromosome 1 from dad. You'll get chromosome 2 from mom, 2 from dad. 3 from mom, 3 from dad. 4 from mom, 4 from dad. So you get the exact same. So you're going to get a chromosome that tells you about hair color from your mom and your dad. Those would be homologues. The definition of a homologous chromosome, what does homo mean? Same. A homologous chromosome is the same chromosome, but came from a different source. It was the same chromosome, but it came from a different source. Same chromosome came from a different source. And so, of course, the source is mom or dad. You can say homologous or you can say homologous. Most of the time I say homologous. I was just trying to sound it out for you. Alleles, so when you have chromosomes, like let's say it's the chromosome for hair. Chromosome hair from mom, chromosome hair from dad. The alleles are the choices you have for that characteristic. So for hair, what are some choices that we could have for hair? Brown, red, blonde, okay? Those are alleles. Alleles are your choices. And again, I'll go into this in another lecture this week. So you'll get allele from dad and allele from mom, and that's where we get into dominant and recessive. Meiosis will separate these two homologous pairs. So as you go through meiosis, so once you've been formed, you're the result of sperm and egg meeting, but you will eventually go through meiosis, and most of you already are currently going through meiosis. When you go through meiosis, if you have a cell that has both sets of chromosomes, so half your mom, half your dad, we call that diploid. These are the majority of your cells. All your somatic cells are diploid, and your stem cells for gametes are diploid. After meiosis, all of your cells are haploid. Haploid means that it has one set of chromosomes. And we talked about the term ploid last week as well. But ploid is referencing a number of chromosomes. So haploid is one set of chromosomes. Diploid is two sets. What's triploid? What's polyploid? Many. And I mentioned that polyploid really only applies to plants and genetically modified organisms. Okay? So this slide right here compares mitosis, which you should already be familiar with meiosis. So in mitosis, we say we have two genetically identical daughter cells in mitosis. In meiosis, we say we have four genetically different daughter cells. Okay? So it mentions that here. In mitosis, all your daughter cells are diploid. In meiosis, all the daughter cells are haploid. Interphase occurs in both. Interphase occurs in both mitosis and meiosis just once. And do you remember what happens during interphase? We have three parts to interphase. G1, what happens in G1? Growth. S, DNA replication. G2, preparation for cell division. Yes. Interphase occurs one time in both of these. Just one time. In mitosis, the correct answer to that quiz question is considered what type of division? It's technically considered nuclear division because the nucleus divides, but if you put cell, I gave you credit. But mitosis is nuclear division. Meiosis, we call it reduction division because it's reducing the number of chromosomes in each cell. Because it's reducing the number of chromosomes in each cell. So mitosis is nuclear division, technically. And meiosis is reduction division. Anyways, back to interphase. Interphase, during S, the genetic material is copied. You started off with the chromosome, but when you copied it, we call it sister chromatids. Okay? And you're going to hear, between these two phases, you're going to hear the same terms again. You're going to hear sister chromatids, you're going to hear kinetochores, you're going to hear prophase, metaphase, anaphase, telophase. You're going to hear all of that again. You need to know, am I dealing with the skin cell or am I dealing with the sperm cell? Does that make sense? You need to know what the question is asking you. If I'm dealing with the skin cell, what phase am I going through? Or what process? Mitosis. If I'm dealing with the sperm cell, meiosis, right? Or meiosis. Either way, however you want to say it is fine with me. Just making sure that you understand which one you're dealing with. If I'm dealing with the skin cell, my product is going to be the same or different? Same. If I'm dealing with meiosis, my product's going to be different. Okay? All right. So in mitosis, which we covered last week, it's interphase is part of the cell cycle, but then prophase, metaphase, anaphase, telophase, and it's over. In meiosis, we repeat it. So it happens twice. In meiosis, it happens twice. So we have meiosis 1, and we have meiosis 2. So what you're going to hear, if it's in meiosis 1, it'll be called prophase 1, metaphase 1, anaphase 1, telophase 1. If it's in meiosis 2, it'll say prophase 2, metaphase 2, anaphase 2, telophase 2. How many times does it go through interphase? Once. Keep that. That's going to be a question. I already know it's going to be a question. Both only go through interphase one time. Meiosis goes through prophase twice, metaphase twice, anaphase twice, telophase twice. But they both only go through interphase once. Okay? So, meiosis. Here, Another thing that I want to point out, and when you're looking at diagrams, this will also help you figure out whether you're dealing with mitosis or meiosis. I started off, and again, for teaching purposes, we're not putting all 46 of your chromosomes up here, because if we put all 46, it becomes very confusing. So we're just pretending like you have one, and what's the shape of your chromosome? You have what type of chromosomes when we talk about shape? Linear. Okay? If it's a prokaryote, what shape? Circular. And how many chromosomes does a prokaryote have? One circular chromosome. Prokaryote. What's the only thing on Earth that's prokaryotic? Bacteria. And it goes through mitosis or meiosis? Binary fission. Trick question. Good thing to test this tomorrow. Not today. Okay, so we have, we're just using one chromosome. You have a homologous chromosome. So you got chromosome 1 from mom, chromosome 1 from dad. Okay? During interphase, those chromosomes are replicated into sister chromatids. Same we've seen before. Okay? Nothing new just yet. Here's the first difference. Crossing over. This is also referred to as recombination. Crossing over is where the chromosomes get really tight together, and we're going to learn that term today, get really tight together, and they just randomly switch genetic material to make each sperm and each egg different. This is why they're different, because of this process right here. But do you notice how these chromosomes are side by side? That indicates to me I'm dealing with meiosis, because if it was mitosis, they would be in a straight line. Okay, if they're side by side, I'm dealing with meiosis. So then, here's meiosis 1. They divide one time. Do they go through interphase again? No. So now all they do is divide again. So these 4 haploid cells at the end are all different. Every single one of them is different. Here was the parent cell that was diploid. Here are the offspring, haploid. Okay? Key distinction here is that the chromosomes are side by side and that they're switching genetic material. That tells me that I'm dealing with meiosis. And meiosis only occurs in gametes, which are produced in the gonads. Yes. Okay? So, prophase 1. Let me just say this. Remember how in mitosis it said prophase, these are all the things that happen. Metaphase, these are all the things that happen. Anaphase, these are all the things that happen. Here, the only thing that's going to be brought up are the things that occur that are different. So, the majority of this stuff happens the same as prophase, metaphase, anaphase, telophase. The differences are pointed out in this chapter. So, during prophase 1, the big difference is that this is where recombination occurs. Recombination is also called crossing over. You need to know both terms. During prophase 1 of meiosis, this is where recombination occurs. I am going to go over synapse and tetrads, but I'm going to do it with a picture. I'm not going to try to explain it just with words. So, prophase 1, big distinction here. This is when recombination or crossing over occurs. This is what makes us genetically different. This is what leads to genetic variation or genetic diversity. Okay? Prometaphase 1, pretty much the same. We attach to the kinetochores and we get pulled to the middle. Where in metaphase we will line up. So what it's showing here, we have all of this occurring. Our chromosomes, our chromatin is condensing into chromosomes. Here, they're forming a tetrad. Because there's 2, and you can't technically see that here, 2 and 2, when they come together, 2 and 2 is 4. So we call it a tetrad. Tet is 4. Tetra anything is 4. So they form a tetrad, and it's going to show you a lineup like this, where they're all next to each other, because they're trying to show you written in like a two-dimensional setting. But technically, the chromosomes line up like this, and because they're so tight and close, they switch genetic material, and that's what this is trying to depict. They're not side by side, they're on top of one another, which is what we call the synapse, or they form a tetrad. Another term you're going to hear for that is bivalent. Bivalent is another term for that tetrad. Okay? So we have them forming synapse, and it's very difficult to see that drawn. Here, still in prophase 1, this is all prophase. In prophase 1 is where that recombination occurs. So crossing over or recombination, this is what allows for genetic variation or genetic diversity. No two sperm, no two egg are the same. And then here takes us into prometaphase. How do I know this is getting me closer to metaphase? Because I can see my spindles. And remember, those are like the fishing line. And some of those spindles are going to make contact and attach themselves to a kinetochore, and we call those kinetochore spindles. And some of them are going to throw out, and they're not going to hit anything, and we call those the non-kinetochore spindles. Do you kind of recall those terms from last week? I hope so. Good. So prometaphase. Oh, also, again, see how these are side by side? They're not in a single file line like they were in mitosis. Those chromosomes are side by side. Okay? Here's talking about metaphase. Metaphase is where they line up in the middle. Metaphase, middle. Okay? During this, they line up in the middle, and that's a checkpoint to make sure that everybody's lined up the way they're supposed to. After metaphase comes anaphase, where they're moved away or apart from one another. If they separate the way that they should, you should have a healthy offspring. But what we see happening is this phenomena called non-disjunction. Non-disjunction says that these two chromosomes that were supposed to separate stuck together. And that causes a problem. Because now, at the end, my daughter cell's supposed to have one chromosome, and how many does it have now? 2. So that means that if that egg or that sperm is used to make a baby, that baby is going to have genetic complications. The prevalency of non-disjunction, I'm going to show you some diagrams, increases with age. So that's why they say people tend to have children with Down syndrome the later on in life that they become pregnant. Because the timing and the occurrence of non-disjunction increases with age. Okay? So most, now I'm not saying you can't be 21 and have a child with Down syndrome, because that happens also. But we see a higher occurrence in people who are older having children and they have Down syndrome. Any other chromosome that they get extra of, it's going to cause them to pass away, so they're selected against. So we see a lot of older couples trying to conceive, continuously miscarry. And it's because there's too many chromosomes. Just Down syndrome is one that you can actually survive outside of the uterus, so you haven't been selected against. Okay? And we'll get there to those later this week. Telophase, same thing's happening. They're on the separate sides. That pinching action begins. Okay? That leads to cytokinesis. So here again, I told you in metaphase, of course, they've lined up. Remember, in prometaphase, if there is a prometaphase, those spindles attach to kinetochores, and then some of them didn't. My non-kinetochore spindles, they're lined up. Again, we don't have a single file line. We have those doubles. Okay? Then during anaphase, the ones that are connected to the kinetochores should pull them apart. All right? The non-kinetochore spindles are going to push, and we talked about that during mitosis. Okay? Telophase, you have your genetic material here, that nuclear envelope, all of that. Okay? And their cells are going to pinch in half during cytokinesis. It mentioned here, there is no interphase. This stuff isn't replicated again. It's just going to go straight into meiosis 2. During meiosis 2, we have prophase 2, metaphase 2, anaphase 2, and telophase 2. And it tells you that. And you can see that pretty much nothing changes. Okay? Because there was no need to replicate that genetic material. All I need to do is separate what's there. All I need to do is separate what's there. In a few slides, just so you know, non-disjunction can occur in meiosis 1 or meiosis 2. Non-disjunction can occur in meiosis 1 or meiosis 2, where those chromosomes stick together. I'm going to show you 2 examples, but I just wanted you to know that there's 2 opportunities for that to happen. Okay? This is going through meiosis using the diagrams, the same setup, following that same setup that we had. This is just meiosis 2. So I have my genetic material. You can see my spindles are forming. During prometaphase, if there is one, those spindles are attaching to the kinetochores. Those that don't attach are my non-kinetochore spindles. So you have that set up there. Okay? Here you have in metaphase. Now they're lined up because I only have half. Okay? Then anaphase apart. They're being pulled apart. The non-kinetochore spindles are going to push. And then here you have telophase / cytokinesis, where I have 4 genetically different daughter cells that are haploid, have half the number of chromosomes. Okay? Meiosis. And that's the way it should happen textbook. Of course, we're going to talk about issues, but any questions so far? Anybody need clarification? Okay? So this is kind of showing you in comparison to mitosis. So in mitosis, it doesn't say prophase 1, prophase 2, any of that. In mitosis, it just calls it prophase, metaphase, anaphase, telophase. All right? Also, here's the key. In metaphase, it's in a single file line. Okay? Same thing happening, but my daughter cells are identical to my parent cell. They're genetically identical. These 2 are identical to each other. They match the parent cell and they themselves are identical. Versus meiosis, where they're double stacked, where we have our tetrad or bivalent, recombination and crossing over occurs to make them genetically different or increase genetic variation. If that's an answer choice, you cannot go wrong with that. That's just one of those biology things. Like structure is function. Structure determines function is a key answer. Genetic variation. In life, all we want is genetic variation. So you can see they're separating. And here is just at the end of meiosis 1. So I still have some sister chromatids there. It's going to go through the whole thing again. But at the end, I have 4 genetically different daughter cells. 4 genetically different daughter cells in meiosis. Any questions on the comparison for mitosis to meiosis? Okay. In meiosis, we reference sex chromosomes. I'm going to tell you this. People tend to think that sex chromosomes are only present in your sperm or egg, and that is not true. I can pull one of your skin cells and look at it under a microscope after, of course, I've extracted the genetic material, and I'm using a high-powered microscope, not just a compound microscope. But your sex chromosomes are in every single one of your cells. So I can find a skin cell and run it through a machine, look at it, and tell you that's a skin cell from a female. Okay? Or that's this from a male. Okay? If they find semen at a crime scene, why do they make the conclusion that it came from a male? Male is the only one who makes. So those are things that we just, understood. But if we find skin cells or hair fibers, you have to analyze them. Are those female or male hair fibers? And then sometimes you find hair at people who have animals. They have like dog hair and stuff. And then people are like, oh, well, that's human. No, you have to look at it, because we know if we look at that, if there's a different number of chromosomes, that it didn't come from a human. So we can exclude a human or a suspect as a result of that. Okay? But every single one of your cells has sex chromosomes. So we know that. But in humans, as well as in a lot of other organisms, which we'll talk about, we have 2 X chromosomes if you're a female. And if you're a male, you have an X and a Y chromosome. If, for some reason, and it does happen, you turn out and you have 3 X's, but there's 1 Y, which means a lot of non-disjunction occurred and we have an issue. We do, but we'll talk about that. If there's a Y chromosome, it is automatically a boy. The Y chromosome is responsible for the development of a male. So if there's a Y chromosome, I know that that individual is a male. It doesn't tell me what type of male, if they have any other diseases, but if there's a Y, I can tell you it is a male. Okay, because that chromosome, its only job is to make a boy, to be like a boy. Okay? So if a female has a Y chromosome, she's not a female. She's a male, because she's got a Y chromosome. Okay? So it's not a thing. All right? Here, it tells you that, what I already mentioned, but here it is in writing, that women, when they make their eggs, every egg is female. Okay? Now, I've heard, like some of my students have said, so you're technically a girl? You're from the beginning, even if you turn out to be a boy? No, because if you were technically a girl, you would become a girl. Okay? You're not a baby if it's just your mom's egg. That's not a baby. That's 23 there's no baby there. Does that make sense? So you can't say, I was a girl before I was a boy. No, you weren't. You weren't. You're an egg, and if that egg becomes fertilized, now we have a baby. And if that egg is fertilized by a female sperm, then yes, you have a girl. But if that egg is fertilized by a male sperm, then you have a boy. 50% of male sperm are female, 50% of male sperm are male. It's just the way it works out. How can you control whether or not you're going to have a boy or a girl? There is some scientific evidence that shows once the female has ovulated, a female can only get pregnant 24 to 36 hours a month. That's it. She could have sex every single day of the month and not get pregnant unless it was during that window. She can only get pregnant during, let's say, 36 hours. Once she ovulates, if she engages in intercourse during that first 12 hours, she'll conceive a boy. If it's the end of that, she'll conceive a girl. Male sperm can operate under warmer temperatures, which is when a female first ovulates, she's running a fever. So her basal body temperature is higher, which is why she feels sad and down and hungry and all that stuff. But after her basal body temperature has come back to homeostasis, the male sperm aren't as successful. So she'll conceive a girl. Okay? So there is a little bit of female control there, but the sperm, the male and female sperm, are made by the male. Here's where it's going to talk about genetic variation. Okay, so here is the crossing over part. It's just going to take it into a little bit more detail so you can physically see what's happening. But as I mentioned, crossing over occurs, or recombination occurs during prophase 1. That's a test question, and that's why I keep saying it. That's a big difference for meiosis. Okay? It mentions that these tetrads, which is a term I've used today, are held together, and it's calling it, instead of a synapse right now, it's calling it a synaptoneal complex. Why? Because it sounds fancier. A synapse and a synaptoneal complex are the exact same thing. Okay? The chiasmata, which, well, that's an anatomy thing too. That's where our optic nerves cross over. Chiasmata just means it's crossing over. So I'm going to show you what that looks like. Okay? Again, these are depicted side by side. they're technically put on top of each other. And when they're put on top of each other, they're held together super tight while they exchange genetic material. So we have homologous chromosomes that were replicated during interphase. So I have sister chromatids right here. Okay? I'm in prophase, so they've condensed into chromosomes. And during this time, they are on top of each other, these homologues. They switch genetic material. This actual spot where they're crossing over is called the chiasmata. That's what that's referencing. Just the physical crossing is called the chiasmata. This increases genetic variation. Once they've switched some genetic material, they'll separate, and now I have two different sets of chromosomes. Okay? They're just genetically different now. This occurs during prophase 1. It adds genetic variety. That is the goal of meiosis, genetic variation. And I talked about the importance of this because if we were all genetically identical and I got cancer, you're all going to have cancer. And if we were genetically identical, I'm a girl. Yeah, you're all girls. So what does that really say for our species? Yeah, it's not a thing. It's not a thing. Okay? So we want genetic variation. Is that it? Yes. Okay, questions, clarification. Yes, sir. Prophase. Yes, sir. Recombination only occurs in prophase 1. Other questions? Anything else I can clarify? Okay. As soon as I get to the house and get connected to Wi-Fi, I will get all these uploaded for you. Good-okey?